Hello, this is Priyanka Arora from Department of Biotechnology, Rama University. Today I am here to give the lecture on molecular biology. The topic of my lecture is DNA replication. So DNA replication is the process of copying the exact copy of the DNA. Before the cell divides, it needs to copy complete genome so that each daughter cell receives the exact copy of the DNA which its parent cells have. Before going into the DNA replication, we will first see the structure of the nucleotide. We know that the DNA, it is a double helical structure. This double helical structure is formed by the phosphodiester bond between two nucleotides. First, this is first nucleotide and the second nucleotide. The bond which is formed between two nucleotides is a phosphodiester bond. So we will see how this phosphodiester bond is formed. The 3' OH of the DNA, the first nucleotide of the DNA, it forms a nucleophilic attack on the phosphate of the incoming nucleotide. It forms a nucleophilic attack. The lone pair which is present in 3' OH, it forms a nucleophilic attack on phosphate of the incoming nucleotide. In this way, this bond the oxygen bonds get destabilized and it breaks this bond, forming a phosphodiester bond. This bond is known as phosphodiester bond. This is a bond which stabilizes the DNA. And this bond, the high energy bond, this is released as pyrophosphate or PPI. It has the anhydride bond. And this anhydride bond is a high energy bond and it gives the energy which is required for the DNA replication to happen. This phosphodiester bond is known as phosphodiester because already there is a presence of monoester bond between the phosphorus of this nucleotide. Okay, so this is how the nucleotide is getting attached in the DNA. This happens in 5' to 3' direction only because it needs a 3' OH, free 3' OH for its elongation. Thus, in this way, the polymerization of the DNA happens. This happens by the presence of DNA-dependent DNA polymerase. DNA-dependent DNA polymerase means the enzyme which uses DNA as its template and it polymerizes the formation of DNA only. Thus, the DNA forms its exact copy during the replication process. This enzyme is having various other functions. First, it is having a polymerization activity, nuclease activity also. Nuclease activity means the breaking of the nucleotide bonds. The breaking of the nucleotide bonds can be exonuclease or the endonuclease. Exo or endo. Exonuclease which is present in the DNA polymerase can also be of two types, 3' to 5' exonuclease activity or 5' to 3' exonuclease activity. The DNA polymerases can have both the functions, 3' to 5' exonuclease activity or the 5' to 3' exonuclease activity. It means it acts on 3' OH and it removes the 3' OH end of the 3' OH end of the nucleotide. 5' to 3' exonuclease means it attacks on 5' phosphate group and it removes that from the end. DNA dependent DNA polymerase also has, in its active site, it also has Mg2 plus ions. It has Mg2 plus ions. These ions are required to overall form salt bridges between the overall negative charge of the DNA. The DNA has the DNA phosphate backbone which is negative charge. The incoming nucleotide also has a negative charge and the DNA polymerase has two of the amino acids in its active site which is aspartate and glutamate which are also full of negative charges. To neutralize the negative charges, the DNA polymerase active site also have a magnesium ions which will form the salt bridges between the negative charges and the magnesium ions to neutralize the overall negative charge. Thus, in PCR, when we perform the PCR in the laboratories, we add Mg2 plus in the buffer to overall neutralize the negative charge of the DNA. Okay, so we will now study what are the different types of DNA polymerases which is present in prokaryotes and what are the different types of DNA polymerases which are present in eukaryotes. Then we will move to what are the different types of other enzymes which are present in the DNA which are used in DNA replication. there are 5 different types of DNA polymerases in eukaryotes, and there are also 5 different types of DNA polymerases in prokaryotes and in eukaryotes. Okay, so if we'll consider the prokaryotes, there are DNA polymerase 1, 2, 3, 4, 5. The DNA polymerase 1, it has 5' to 3' polymerization activity. 5' to 3' polymerization activity means it will elongate the nucleotide in the 5' to 3' direction. So this is my DNA. This is the primer which is added. It will add nucleotide in 5' to 3' direction. So it will move forward. It will add the nucleotide in this direction. Okay, the 5' to 3' exonuclease activity is known as primer removal activity. Exonuclease activity means it will remove the nucleotide bond, it will remove the nucleotide base from it. So 5' from the 5' end, it will remove the nucleotides. This is only required if we have to remove the primer. So 5', 3' exonuclease activity is known as primer removal activity, and 3', 5' exonuclease activity is known as proofreading activity. Proofreading activity means if by chance there is a mispriming, suppose in the template there is a presence of A, and while adding a nucleotide base, instead of G, the nucleotide DNA polymerase added nucleotide C. There will be a distortion in the pocket which is present in the DNA polymerase, and it will the distortion will lead to the breakage of this bond in the 5' to 3' direction. It will check, it will lead to the breakage of this bond. This nucleotide will be removed. It will again go and look for the correct nucleotide, and it will add a correct nucleotide here. So this activity is known as exonuclease activity, 3' to 5' exonuclease activity, or the proofreading activity. So the RNA polymerase, the polymerizing activity is present in all the polymerase enzyme, because this is a major activity of the polymerase. So it is present in all the polymerases which is present in eukaryotes or in the prokaryotes or in the eukaryotes. Okay. So the primer addition activity, the primer removal activity, is present only in DNA polymerase 1. The primer removal, this primer which is added with the help of enzyme primase, is removed in prokaryotes with the help of DNA polymerase 1. The exonuclease activity is present only in DNA polymerase 1, 2 and 3. Rest 4 and 5 do not have any exonuclease activities. In the eukaryotes, the DNA polymerase alpha is involved in priming activity. It is responsible for addition of the primer. This addition of the primer is happening because of 5' to 3' polymerizing activity. Thus, it does not have any exonuclease activity. DNA polymerase beta is not responsible for any exonuclease activity, and it is only involved in DNA repair, especially in the base excision repair. DNA polymerase Dept, this gamma, is responsible for polymerizing activity, and it also has 3' to 5' exonuclease activity, and it is involved in elongation of mitochondrial DNA. It happens in the mitochondria. The replication happens in the mitochondria with the help of DNA polymerase gamma. Rest epsilon and delta, these are the main polymerase enzymes in the eukaryote, and it has proofreading activity. It does not have primer removal activity. Primer removal activity in eukaryotes does not exist in any of the DNA polymerase enzymes. It happens by other procedure. Okay. So next enzyme after DNA polymerases, first of all, DNA polymerase enzyme in E. coli was first studied by Kornberg, K-O-R-N, Kornberg. So next is, because we know the DNA polymerase can add only in 5' to 3' direction, because it needs free 3' OH, because it needs free 3' OH, thus it needs a primer to start its activity. The primer in prokaryotes are added by the enzyme primase, and in eukaryotes the primer is added by DNA polymerase alpha. Primase in prokaryotes and DNA polymerase alpha in eukaryotes. It adds the primer which is responsible, which gives free 3' OH, and it helps in elongation of the nucleotide for the replication to happen. Okay. So all the other enzymes, which are required first is the processivity of the DNA. The processivity of the DNA polymerase is very less. The processivity of the DNA polymerase in prokaryotes, it is 1 nucleotide per second. It is a very slow speed for a nucleotide to add next nucleotide. Thus, if it will take so much time, then the replication is going to take a lot of time. So there is a to increase the processivity, there is a clamp load. There is an enzyme which is known as clamp. This clamp does what? It holds the DNA in its position with the help of, it is a ATPases, it holds the DNA in position with the help of clamp loader, and it does not allow the DNA to fall off and increase the processivity before it gets released off from the DNA, and next time it gets added, thus reducing the time of the DNA replication. Clamp and clamp loader are also important enzymes for DNA replication. Other enzymes are helicases. Helicases are the enzymes which breaks the DNA hydrogen bonds between the two nucleotides. The two nucleotides, this is also known as unzipping enzyme. It unzips the two double helical structure of the DNA, and it unzips the two double helical structure of the DNA, makes the single strand available for the replication to happen. Next, the enzyme which is important is DNA topoisomerases. When the DNA is uncoiled from between with the help of helicases, there is overwound found in ahead of the replication fork. To release this overwounding or the underwounding, there is an enzyme which is important, which is known as DNA topoisomerases. DNA topoisomerases are of various types, and these are also known as DNA gyrases. The other enzymes which are required in DNA replication are the SSBP protein. SSBP protein is single stranded binding protein. Since after the action of helicases, there is a presence of single strand. The single strand, the hydrogen bonds between the two strands, they tend to form again. To avoid this resealing of the DNA helix, there is a presence of single stranded binding protein. These single stranded binding protein, they binds to a single stranded DNA helix, and it will not allow the resealing to happen. So these are the enzymes which are required for DNA replication to happen. Now we will see what are the enzymes, which of these enzymes are present in eukaryotes and which are the enzymes which are present in prokaryotes. So in prokaryotes and eukaryotes, the different enzymes have different names. The helicases in the prokaryotes are known as DNAB, whereas in prokaryotes it is MCM. The SSBP protein in prokaryotes is SSB, that is single stranded binding protein, whereas in eukaryotes it is replication protein A or the RPA. The primase in prokaryotes is DNAG, and in eukaryotes it is DNA polymerase alpha, as we have seen in DNA polymerases. The replicase is DNA polymerase 3 in prokaryotes and DNA polymerase epsilon and delta in eukaryotes. The topoisomerases are gyrases, that is topoisomerase type 2 subclass in prokaryotes, and topo 1 and 2 in eukaryotes. The processivity component is beta clamp in prokaryotes, whereas in eukaryotes it is PCNA, that is proliferating cell nuclear antigen. In the clamp loader in prokaryotes is gamma complex, and in eukaryotes it is RFC, that is replication factor C. The primer removal in prokaryotes is done by RNA polymerase 1, and in eukaryotes it is done by RNase H, FEN endonuclease. So these are the enzymes which are involved in DNA replication. By this we end the DNA replication, the enzymes which are required in DNA replication. There are three processes by which the DNA replication continues. That is, first the initiation happens, then the elongation, and the last is termination. There are different steps, different mechanism by which the DNA replication proceeds in eukaryotes, and different methods in which the DNA replication proceeds in eukaryotes. So in next lecture we will see the initiation, elongation and termination in prokaryotes.